obviously in 100 BC. That's beyond their scope of understanding. So then we also have Aristotle, and I'm kind of jumping around a little bit in terms of time. He was in the 300s BC, and most people think of Aristotle as a great scientist slash philosopher and so forth. But we can kind of credit Aristotle for holding up the advancement of microbiology. So Aristotle believed that living things arose from 3 different processes. And those processes were sexual reproduction. Let me get another marker. This one's not working great. Sexual reproduction. This one's not that much better. Asexual reproduction and spontaneous generation. So both sexual reproduction and asexual reproduction are a form of biogenesis, meaning that living thing comes from another living thing. Okay? So sexual reproduction is a process whereby the offspring have a combination of genetic makeup from both of their parents, and it leads to genetic variation in the offspring. So for example, you don't look exactly like your mom or dad, and you also don't look exactly like your siblings, and that's due to this process, which of course nobody wants to think about, thinking about how they came to be, but in terms of parents. But genetic variation, that's the key, that's the result of sexual reproduction. Asexual reproduction, there's no genetic variation, and this is how bacteria reproduce. Basically, bacteria clone themselves, right? So they do reproduce, so it is a form of reproduction, but it's not sexual. There's no exchange of genetic material and variation in the offspring, okay? The offspring have the same genetic material as the parent, okay? So that's asexual. But both are living things leading to more living things, okay? Spontaneous generation is a form of abiogenesis. Anytime you put an A in front of the word, it negates it. So sexual reproduction, asexual means it's not sexual. Biogenesis versus abiogenesis, abiogenesis means it's not biogenesis. So basically, spontaneous generation is the formation of living things from non-living things. And so Aristotle believed that that was one way in which living things arose. And so remember, I said they didn't have a lot of tools of science back then. And so they made a lot of observations. So if you were a scientist back during 300 BC or whatever, and you saw dry dirt and no frogs, and then it rained and you saw a big mud puddle and now you see frogs, where do frogs come from?
They would think the mud.
Yeah, from mud. Frogs come from mud. So that would be a living thing coming from a non-living thing. We know differently, but again, they had no tools of science. They just made these simple observations. Just for fun, I have 2 recipes for you in case you ever want to make bees or mice. I have spontaneous generation recipes. You don't need to know this. But bees, you kill a young bull and bury it in an upright position so that its horns protrude from the ground. After a month, a swarm of bees will fly out of the corpse. That's how to make bees. If you want to make mice, you don't get other mice to make mice, at least not according to Aristotle. You place a dirty shirt or some rags in an open pot or barrel containing a few grains of wheat or some wheat bran, and in 21 days mice will appear. So that's how you make mice. Okay? They just spontaneously generate. Okay? So this really held back the advancement of microbiology because we know that microbes come from other microbes, and illness comes from microorganisms, at least infectious illness, right? So colds and cuts that become infected, they come from microorganisms. They make you sick, right? Or they can make you sick. According to individuals who believed in spontaneous generation, that's not how you get sick. There are no microorganisms that cause disease. Disease comes from things like bad air. And they even had a term for that. They called it miasma. Just meant bad air. And so that's why people got sick, for reasons like that. But not because there was a living organism that was, living inside of you, making you ill. So it really held up the belief in micro for thousands of years, actually, for a very, very long time. So I'm going to jump around in time just a little bit. I really don't have many dates between 100 BC and, say, the 1600s, because there really wasn't all that much going on in microbiology till then. So, but once I get to the 1600s, I will jump around a little bit in time. So I want to show you Robert Hooke's cork section. So Robert Hooke, he was in the, late 1600s, early 1700s, and he built himself a microscope. And he used that microscope to observe thin slices of cork. I don't know that that's cork on this side, but this is definitely cork over here. And what he did was he was looking at these very thin slices of cork, which come from the bark of trees, and he noticed these little compartments. And he thought those compartments in the cork looked very similar to the rooms that monks lived in. Anyone know what you call a room that monks live in? A cell. And so he called these cells, and the name stuck. So we can thank Robert Hooke for calling them cells. Now, that was his contribution. Without him calling them cells, we wouldn't have something called the cell theory, which came a couple hundred years later. And the cell theory is credited to Schleiden and Schwann in early 1800s. One of them was a zoologist and one was a botanist. Don't ask me which is which. I don't know, I don't remember, and it's not that important, although it's probably important to these two guys. But they were botanists and zoologists, and they developed what was called the cell theory, which had three tenets, three statements that made up that cell theory. All living things are composed of cells, which we kind of talked about a little bit when we were talking about the differences between bacteria and viruses. Viruses aren't composed of cells. Cells are the smallest unit of life. So we're composed of millions and millions of cells. How many cells are bacteria composed of? One. That's the smallest unit of life. Nothing smaller than a cell is considered alive. And then all cells come from pre-existing cells. What does that statement go against? Spontaneous generation. Now, officially, this cell theory came out before spontaneous generation was completely disproven. So these guys were a little ahead of their time. But during their time, there was a lot of work trying to disprove spontaneous generation. Then we have Anton van Leeuwenhoek. Here's his name. It's kind of a crazy name. He was a Dutch cloth merchant, and he was also, I think he was around, he was in the 1700s mostly, late 1600s, early 1700s. I think he was roughly around Robert Hooke, around those same times. And he made his microscope very different from that of Robert Hooke's. The interesting thing was he was able to magnify things 300 times, which was very technologically advanced for that time period. I don't believe that Robert Hooke's could magnify as much. His didn't really look like what you would call a microscope, but it looked kind of like this. And you can see it's really small. This is a centimeter ruler, and it's maybe less than 9 centimeters long. It's a really small thing. But he was a cloth merchant, but in his spare time he liked to grind lenses. And so he made these teeny tiny little lenses that he put in his microscope. And they were the best lenses around. Nobody could make lenses like Anton van Leeuwenhoek. And he was able to see all kinds of things under his microscope. He would look at samples of like mud puddles, and he would look at saliva from people who were ill and all kinds of stuff. And he would see these things in his microscope, and he didn't know what they were, so he called them animalcules.
Bless you.
And animalcules, the word is kind of a mixture of animal and molecule. He wasn't really sure what he was seeing. But he was seeing microorganisms. Were they bacteria? Probably not. They were probably other microorganisms that were larger, because at 300x, bacteria would be just teeny tiny little pinpoints. But they were microorganisms of some sort. So this was great, because once you can see something, you can study it a lot better. The only problem was, when he died, he took the secret to making these great lenses to the grave with him. And it held back the advancement of microscopy for like 100 years. Right? Now, the big problem with the advancement of micro was still spontaneous generation. But microscopy, he kind of, he didn't share. So he should have shared. So, like I said, in the 1800s or so, even a little before, late 1600s to 1800s, scientists were starting to really question spontaneous generation. And so they started doing experiments to try and disprove its existence, because the belief in spontaneous generation was common knowledge. It was the same to them as us thinking that the earth is round, right? We know that the earth is round. Well, they knew that spontaneous generation existed. And so, good scientists were trying to disprove it. So back in the mid-1600s, Francesco Redi, who was an Italian physician, did this experiment, because everybody knew that flies and maggots spontaneously generated from rotting meat. They didn't come from other flies. They came from rotting meat. So he didn't think that was true, so he set up this experiment. He got 3 jars. He put rotting meat in all 3 jars. He covered the one, sealed the one, left one open to the air, and then put gauze or like a cheesecloth over top of the 3rd one. And exactly what you guys would expect to happen is what happened. What believers in spontaneous generation would expect would be that you'd have flies and maggots on all 3. Okay? But what happened was exactly what we would think. The one that was sealed, nothing. The one that was open, flies and maggots. Right? The one that was covered with gauze, there were flies and maggots, but not on the meat. It was up here. They couldn't get in, so they weren't generating from the meat. And he thought this disproved spontaneous generation. So the critics to him, the ones that really believed in spontaneous generation, said, well, we'll give it to you for larger things like, flies and maggots, but this whole microorganisms causing disease thing, we don't buy it. We don't buy it. So it kind of helped but didn't solve the problem. Then came Lazzaro Spallanzani. He was about 100 years after Redi. He was also, as you can tell from his name, also Italian. And he set up a different experiment using broth. So broth, you can kind of think of as, it is a liquid medium in which you grow bacteria. Basically, it's a liquid that has lots of nutrients in it. Bacteria like to grow in it. And when bacteria grow in a broth, the broth becomes cloudy. And we have a term for that. Anyone start their lab reading and know what term we use instead of cloudy?
Turbidity.
Turbid, right. Turbid. So it becomes turbid. You see turbidity. And the more turbidity you see, the more bacteria are growing. Okay? So there's definitely a positive correlation between how much turbidity you have and how many microbes are in the broth. Okay? So what he did was he actually set up 2 flasks. Okay? One was open and one was sealed. And he boiled both of them. Why do you think he boiled both of them?
To get rid of the harmful microbes.
To get rid of not just the harmful, any microbes. Right, he was sterilizing it. so that both the control and the experiment were being treated exactly the same, and we were starting basically at ground zero with both. They were both sterile, okay? So he boiled them both, so they were both sterile, and then he just let them sit, right? And the one that had been boiled and sealed, nothing grew. The one that was open, eventually you had microbes growing in it. Did they spontaneously arise? No, because if they did, they would have spontaneously arisen in this one as well. There's dust in the air. There's all kinds of ways in which microbes could actually get in, but they don't just spontaneously arise. And he thought he did a good job, and he disproved spontaneous generation. Critics said, no, you boiled off the life force. So at the time, life force equates to what we would call oxygen, right? They didn't really know what oxygen was back then. You boiled off the life force, and because it stayed sealed, no more life force could get in, and so even though they felt that the microbes spontaneously generated from the broth, because there's no life force, they didn't generate, okay? So this was not sufficient to disprove spontaneous generation. So we're going to take a little side detour for just a second before we go on to the experiments that directly tried to disprove spontaneous generation. This picture is of a bacterium called Vibrio cholera. See if this mark will work. Ah, there we go. So just a couple things before we get started. If you have me for lab, we've already talked about this. When we write the names of bacterial organisms, or even fungi or protists, or even animals, the scientific name is composed of a genus name and a species name. The genus name is always capitalized. Species name is never capitalized. If you're handwriting it, it's underlined. If you're typing it, what do you do? Italicize. If you refer to this organism more than once, what can you do?
Abbreviate it.
Okay, so when you abbreviate it, it's still a capital V, and then you write out the whole species name in lowercase, and you still underline or italicize based on whether you're typing or handwriting, okay? All right, so that's the way I expect you guys to write these organisms' names out. Another thing is, we never call an organism by just its species name, right? And you guys will try and do that in the lab, and I'll get on you for that. There's a reason for that. The species name is very descriptive of the species. However, oftentimes species names are used for multiple genuses. So for example, with bacteria, I can tell you that if you were to say, that pneumoniae organism, I'd look at you and I'd say, okay, Klebsiella pneumoniae, Mycoplasma pneumoniae, Streptococcus pneumoniae, which one are you talking about? So you always have to say at least the genus name, preferably if you're talking about one specific type of organism, you say both, okay? Now, Escherichia coli, we have kind of abbreviated it in terms of when we're talking about it, we just call it E. coli, right? But that's this abbreviation. Most people know that one, but if you want to be technical, first time you write out anything about E. coli, you have to write it out Escherichia coli. Right? Then you can abbreviate it as E. coli. Okay? Make sense?
Mm-hmm.
All right, so this is Vibrio cholera. Vibrio is also a descriptive term. Vibrio means curved. And Vibrio cholera is a bacterium that is a curved rod. You can see these cells are kind of unique. They're curved. Okay? So it gets its name from that. What do you think Vibrio cholera causes? Cholera, right? Causes cholera. So if you're not familiar with cholera, it's a really nasty infection. It causes very nasty, watery diarrhea. And I'll warn you, we talk a lot about diarrhea in this class, just because microbes cause a lot of it. Causes very nasty infection. A lot of people that die from cholera die because they're dehydrated. They lose so much fluid, they can't replenish it. So that's Vibrio cholera. It normally lives in salt water, the bacterium does. However, it can contaminate freshwater supplies, and that's when you have outbreaks. Today, we see them a lot of times in third world countries. We see them in countries who've had natural disasters, and the water supply gets contaminated and you end up with outbreaks of Vibrio cholera. So back in the 1850s, a physician by the name of Dr. John Snow, no relation to Game of Thrones. Maybe they named the character after him, I don't know, but no relation. Dr. John Snow was a physician in London. England in the 1850s, it was the Industrial Revolution. Were there a lot of good sanitation practices during that time period? No, pretty nasty, right? And remember, this is a diarrheal illness, right? So nasty sanitation. So you've got this bacterium, it's causing all these outbreaks in London. You know, the streets are dirty, there's sewage running, it's a nasty place to be, right? And he noticed this outbreak of cholera in London. And he decided he was going to try and figure out what was causing it. So he later became known as the father of epidemiology, and is credited with carrying out the first epidemiological study. So he was trying to figure out not only how they were getting sick, but what was actually making them sick. So he went and he interviewed the people that were sick with cholera. He interviewed family members of people who had died from cholera. And he was trying to find something that they all had in common, right? And he cultured their stool samples. And in the samples, he found that there was this weird curved bacteria that wasn't in the stools of healthy people. And what he ended up finding that they had in common was that all of them were using water from the Broad Street pump. So he went to the Broad Street pump and he cultured the water. And sure enough, he found the same curve-shaped bacteria. So he made a link between the two. They either shut down the pump or they cleaned it. I'm not sure exactly what they did, but once it was identified that it was coming from the pump and people knew that, the illness stopped. Okay? So that's why he gets to be known as the father of epidemiology. You would think that that would also be sufficient for denying spontaneous generation. They weren't getting sick just because of the air was bad. They were getting sick because there were microorganisms in the water that they were drinking, and he actually made that link. But the critics, the ones that still believed in spontaneous generation, said, yeah, no, we still don't believe it. So it still wasn't officially disproven at this point. Any questions so far? Hands tired yet? Great. Great. So it wasn't until this guy came along till we actually had spontaneous generation disproven. So this is Louis Pasteur, and he was a Frenchman, and he was born in the early 1800s. I think it was like 1822. Most of his work was done in the mid 1800s. And so Louis Pasteur did a lot of good. So he actually disproved spontaneous generation in the early 1860s. And then starting in about the early to mid 1870s through 1917, that little window of time became known as the golden age of microbiology. And the golden age of microbiology was a time where there was rapid advancements in the field of microbiology. Remember, there really wasn't all that much going on for thousands of years. And all of a sudden, after spontaneous generation was disproven, There's a short window of time where there's a lot of stuff going on. Why do you think it ended in 1917? Any history buffs? World War One it started.
like
It was
1914.
14 to 18, was it, or 14 to 17?
14 to 17.
14 to 17. So, this was during the time of war, and, the world had other things on their mind, so to speak. So that was about when it ended. There's been a lot of advancements, obviously, recently, but that was a big time, considering there wasn't a lot known before then. So he disproved spontaneous generation about 1860, 1861, and him as a person, he actually was buried by France as a national hero, which not too many scientists get buried as national heroes. Part of the reason for that is because he didn't just like to solve scientific problems just for the sake of it. He was trying to help France. When they had problems, he used science to try and solve those problems. Okay? And so he actually saved the French government a lot of money with a lot of his discoveries. So we're going to start first with how he disproved spontaneous generation, which is going to be this picture down here. So he used what was called the swan neck flask experiment. So it was similar to Spallanzani's in that he used a flask and he used broth, and he was looking for turbidity, okay? But the difference is, first of all, the shape of the flask. When he first started using the flask, it was shaped like this, okay? Like that, like a round bottom flask with this long skinny neck. And he poured the broth in it, okay? You can see them doing that in the first step on the left. They poured the broth in. And then they did two things. They boiled this broth, just like Spallanzani did. Boiled the broth so that there were no microbes in it. You're starting off sterile, right? But they also heated this long, skinny, straw-like neck, which was all glass, by the way. And if you heat glass, it's bendable. And so they were able to bend it into this shape, okay? So now you've got the sterile broth in there, right? Because you boiled it. And he just let it sit. And it never became turbid. Why don't you think it became turbid? Any ideas?
Because it was exiting out, because of the shape, it was kind of just going out into the air.
What was going out?
Microbes.
No.
Because of the vent shape, the microbes couldn't go up the tube, so
Right,
they couldn't.
the microbes couldn't get in. It wasn't they couldn't get out, they couldn't get in, okay? Think of this kind of like the trap in your drain, right? It works the exact same way as the trap in your drain if you look under your sink. The microbes could get in, they could only get to here. They couldn't get up and around and down. So they were trapped, they couldn't get in, and this stayed sterile, okay? Now, he took one of them and he tilted it. And he tilted it so that the broth went up through and into here, right? And then he tilted it back. What do you think happened after he did that?
They collected turbid.
It became turbid, right? Because what had happened was when the broth hit this area, picked up microbes, brought it back into the round bottom flask area. This was sufficient to disprove spontaneous generation. Remember, this was open. With Spallanzani's, he sealed it. Remember, he had that cork in the top. This is left open to the air. So there was no life force driven off, okay? And this was sufficient. The supporters of spontaneous generation said, all right, you got us now. You got us now. You finally disproved it. Interesting thing is there's still at least one of these on display at the Pasteur Institute, and it's still sterile from 1861. So that's kind of crazy.
How's it still sterile?
Microbes couldn't get in.
Oh, it's not the broth in it.
Still has the broth in it.
I thought you meant like the tube. I was like, by now,
What tube?
swan part of it.
Oh, well, this most likely has stuff in it. But again, it can't get into here, so that broth stayed sterile that long. Make sense? Yeah. So that's all it took. So that was a pretty simple experiment, very similar to Spallanzani's, but the difference being that this remained open the whole time, where Spallanzani sealed this, okay, which, got rid of the oxygen. And then this is just a quote by Louis Pasteur: "Life is a germ, and a germ is life. Never will the doctrine of spontaneous generation recover from the mortal blow of this simple experiment." He had a little bit of a chip on his shoulder, it sounded like, but he was right. It never did recover. Just a little bit more about his personal life. He suffered through his life with strokes, had partial paralysis for part of his life as a result. He also lost 3 daughters to infectious disease, so some of the same types of infectious diseases that he studied throughout his life took the life of some of his children. So he had a rough life in some ways. They built the Pasteur Institute in Paris for him, which is kind of a big honor to have an institute built for you because you were so famous in your country. And when he died, he was eventually buried at the Pasteur Institute. So he did a lot of stuff. So we're going to go over some of the stuff that he actually did. One thing I wanted to mention besides disproving spontaneous generation is what he called the germ theory of disease. Basically, this is just a statement whereby he said that disease is caused by microorganisms. Okay? He was partially right. Infectious diseases are caused by microorganisms, but not all diseases. We have genetic diseases. We have autoimmune diseases. We have, all kinds of diseases. Not all of them are caused by microorganisms, just the infectious ones. Okay? So we're going to get started with some of the other things that he did. We're going to start with these 2 nice glasses of wine. So again, he did a lot of stuff that was important to France. France is known for their wine industry, and the wine was not tasting good during Louis Pasteur's day. It was soured, and they called upon him to see if he could fix it. Why is the wine tasting so bad? So he did a couple things. So 2 really important discoveries came out of the wine industry calling him in to help. First one was he figured out what was going on with wine production, how it happened. So he took good wine and he looked at it under a microscope, and he saw these nice round cells. Some of them were budding. They looked like that, meaning they were reproducing. Right? Saw these nice cells. These were yeast cells. Yeast are not bacteria. Yeast are actually in the kingdom of fungi. And yeast cells are actually more closely related to us than they are to bacteria. Okay? They're eukaryotic organisms. So in the good wine, we saw the yeast. So just like you and I need to make energy, make ATP, so do yeast. They do it a little bit differently though. Okay? We carry out what process to make ATP?
Cellular respiration.
Cellular respiration. Aerobic cellular respiration, meaning we use oxygen as the final electron acceptor. Right? Yeast don't need to use oxygen. Okay? So when we do it, we use oxygen, and what's the other thing we need? Glucose. We need sugar. We need a sugar. Okay? We get that from the foods we eat. So yeast, they don't need the oxygen, right? But they need the sugar. Where do you think the yeast get the sugar to make ATP, to make energy, if they're in the wine? The natural sugars from the grapes, right? So they use those sugars in the grapes, and they use them to make ATP, to make energy? Why do they want energy? To reproduce, to carry out all their cellular functions, okay? In the process of making ATP, there are byproducts formed. Those byproducts are carbon dioxide and alcohol. The yeast don't care about these two. They're waste materials. Carbon dioxide and alcohol, waste materials. They don't want them. They don't care about them. All they care about is making energy in the form of ATP. Okay? A winemaker could care less about the ATP. They care about the CO2 and the alcohol. Okay? So this is what Louis Pasteur figured out. And the process became known as fermentation. Right? Which is different from anaerobic respiration. Anaerobic respiration is more like aerobic respiration, but without the oxygen. This is a very different process. Okay? Aerobic and anaerobic respiration fully break down the sugar molecule. So the sugar is a big molecule. When you break it down, you're extracting energy from the chemical bonds. Okay? When you completely break it down, you're getting a lot more energy out of the molecule. With fermentation, you're only partially breaking down that sugar molecule, so you're only getting a little bit of energy out of it, okay? So, for example, in aerobic respiration, which is what we carry out, and some bacteria carry it out as well, you get somewhere between 36 and 38 ATP per molecule of glucose. Right? We would get 36, bacteria would get 38, okay? With fermentation, for every 1 molecule of glucose, they only make 2 ATP. So it doesn't make nearly as much energy, okay? But it works for the yeast, right? It works for them. So the yeast, they want the energy. The winemakers want the CO2 and the alcohol, okay? So what he noticed was that was in the good wine. But in the bad wine, he was also seeing little bacterial cells in addition to the yeast. Okay? Now, like I said, bacteria can carry out aerobic respiration, like we do. But bacteria can also carry out fermentation. They can do either. Okay? We can do either. If you think about it, right? Our muscle cells can carry out fermentation. Different type, it's lactic acid fermentation. But can carry out fermentation when there's no oxygen around. But the bacteria also can carry out fermentation processes. So what was happening was you had these bacterial contaminants in the wine, and what had happened was the bacteria were competing with the yeast for the sugars. Okay? And when bacteria carry out fermentation, they too are making 2 ATP for every molecule of glucose. They also have byproducts. Not always, but usually the byproducts from bacterial fermentation are acids, okay? Not alcohol and carbon dioxide. Usually it's acids. And we're talking about acids like lactic acid or even acetic acid. Others as well, but those are 2 of the most common ones, okay? Anyone know what acetic acid is? It's vinegar. Do you want your wine tasting like vinegar? No, that's nasty, right? That's nasty. So as the bacteria and the yeast are competing for the sugars, not only is alcohol and CO2 being formed, but also some of these acids which are making the wine taste nasty, okay? So that's what he figured out. So now he needed to solve the problem. So what he did was he took the grape juice, before it was wine, just when it was grape juice, and he gently heated it. Okay? He did not boil it, he just heated it to the point where if there was any bacterial contamination in the grape juice, the bacteria were killed. Okay? So he killed off the bacteria, and then they added the yeast to allow for fermentation to take place. process later became known as. Right, pasteurization. And of course, it's not limited to just wine. We carry out pasteurization on lots of different things. Okay? So it's usually a gentle heating, not boiling. There's some exceptions which we'll talk about in Unit 2, but a gentle heating, and it's just sufficient to kill those contaminants that are in the grape juice prior to adding the yeast. If you were to add the yeast first and then heat it, you'd kill the yeast as well as the bacteria. Okay, so you add the yeast afterwards. Okay? Make sense? Is it warm in here or is it me?
Mm-hmm.
I'm cold.
Really?
Yeah.
Maybe you're under a vent, because it's hot over here.
Yeah,
Well,
it is hot.
no, maybe, I don't know. Maybe you're having cold air blow on you, but over here it's hot. I wish. All right, so there's the wine. So what else did he do? Well, we've got this little guy here. So this is a silkworm. So if you weren't aware, silk comes from a worm. Silk's, or silk is made by these little silkworms. So another thing that France is known for is their, their fancy fabrics. What's the word? Cloth. Fabrics. Maybe that's the word I'm thinking. Textile. Yeah, you know what I'm talking about. So they're known for that, and so the silk industry was having some problems. The silkworms were dying, and if they're dying, they're not going to make any silk, and then you're going to have problems making these nice cloths and fabrics and so forth. So they called upon Louis Pasteur, asked him to come help them, and he said sure. And what he was able to do was separate the sick silkworms from the living one, or the sick ones from the healthy ones. And then he was able to actually identify a couple of different silkworm pathogens that were causing the disease, isolate them away, so that only the healthy silkworms were being used. And then no longer did the disease spread. And then we had nice, happy silkworms and more silk being produced. So that was his role there. And then the last thing we're going to talk about are a couple of different vaccines that he developed. So we're going to talk about the chicken, we're going to talk about the sheep, and we're going to talk about this little boy. So we're going to start first with the chicken. So he worked in his lab with what's called fowl cholera. Okay? Fowl as in like chickens. Now we talked about human cholera, and we said it was caused by a bacterium called Vibrio cholera, right? Different organism causes fowl cholera. I don't know what they called it during Pasteur's day, but now we call that bacterium Pasteurella paucita. And that organism causes disease, causes cholera in chickens. So what he was doing in the lab was he was taking the chickens, injecting them with a virulent strain of Pasteurella multocida. Virulent meaning very strong and able to make the individual sick. Okay? You'd inject them with the virulent strain of Pasteurella multocida, and within 48 hours the chickens would die. Okay? So he was working with them in the lab for I don't know how long. So summer came around, and they kind of shut down the lab for the summer. I don't know what they did with the chickens. Somebody must have taken care of them. But they didn't really do much with the bacteria. So bacteria are just like us in certain ways. They need nutrients, just like we need food, and they have waste products, just like we do. Just like we can't live in our own waste, bacteria can't live in their own waste either, okay? So when we grow bacteria in cultures, if you just let them go, indefinitely, they're going to die. They're going to run out of nutrients, and they're going to be living in their own waste, and it's going to kill them, okay? So what we do in a lab is we subculture our bacteria routinely. every X amount of days. And we basically, what we do is we take a small inoculum of the organism from the culture, and we transfer it to new media that is fresh, has no waste materials, has lots of nutrients in. And then the bacteria start to grow really well again. And then after a period of time, you do it again. And this series of subcultures ensures their survival, okay? So if they shut down the lab, they weren't subculturing their bacteria, and it started to weaken. You know, as humans start to die, they weaken. As bacteria start to die, they weaken. And when they weaken, they become less virulent. In other words, less likely to be able to cause disease, okay? So he didn't really know any of this was going on. A lot of things in science are accidents. So came back to the lab at the end of the summer or after his vacation or whatever, and he took that Pasteurella meltosida, which he didn't know had been weakened, and he injected it into the chickens. And what do you think happened? They survived, right? They survived. And he thought that was kind of interesting. So then he took some of the bacteria and started to subculture it again. And you can take a weakened strain, and if you start to subculture it again, over and over again, it will then become virulent again, okay? So then he took the virulent strain and he injected it into some chickens, and they died. So he knew that it was virulent again. He took that virulent strain and injected it into the chickens that hadn't died from the non-virulent strain. And what do you think happened? They survived. They did not die. So this became the first vaccine for chicken cholera. Okay. So why do you think they didn't die? Right. They knew how to fight it off. Their immune system was working. They produced antibodies and memory cells and all this good stuff that we're going to learn about later this semester. And when they saw the organism, the virulent strain, they were able to fight it before it could make them sick and kill them. Okay? So that's what he did with the chickens. So then he thought, well, that's interesting. Let's see if we can reproduce this with something else. So France was also known for their livestock. They had a big agriculture, what do you call it? Industry. That's the word I'm looking for. And lots of sheep and cows. And in France, there was a lot of problems with a bacterium called Bacillus anthracis. Anyone know what this bacterium causes? Anthrax, right? So lots of sheep and cattle would die from this bacterium. So he decided he was going to do an experiment. And he was going to have it publicized. He wanted people to know about this big experiment he was going to do. And so what he did was he took 50 sheep, okay, took 50 sheep. 25 of them were his controls, and 25 of them were his experimental sheep. Okay? So, to his experimental sheep, he first injected them with a weakened strain of Bacillus cereus, or I'm sorry, not Bacillus cereus, Bacillus anthracis. Okay. And the controls, he did not do this to. Okay, he did not inject them with the weakened strain. Okay. Then all 50 of them, he injected with a virulent strain. Now, again, this was publicized, and he said, if this works, my my experimental sheep should survive and my controls should die, right? And that's exactly what happened, okay? So this became an anthrax vaccine for livestock, and it started being used on sheep and cattle in France. Within 10 years, about 3.5 million sheep were vaccinated, and about 500,000 cattle were vaccinated. And the savings to the French economy were so great from saving all these cattle, all these livestock's lives, that they were able to repay their war debts to Prussia with the savings. So that's a lot of money to the French economy. Probably part of why he was buried a national hero, right? So that was his work with anthrax. And then this last picture here, he was working on a vaccine for rabies. And rabies is a viral disease caused by a virus. Nice thing about a lot of viruses, not all, but a lot of viruses, the name of the virus also is the name of the disease. So in the case of rabies, that's the name of the disease. The name of the virus is also rabies, which is nice. So rabies is very, very deadly. So I don't know the exact numbers today, but just a couple years ago, probably only about 10 people ever have survived clinical rabies. So once the symptoms start, you might want to write your will, because if you get to that point, it's not looking good. About 10 people ever have survived clinical rabies. Now, if you get bit by a rabid animal, and nowadays we're lucky we have a vaccine, that vaccine is good if you've been bitten but the symptoms haven't started. If that happens, you get the vaccine, you don't develop rabies, you survive. So, yeah.
Isn't it like 24 hours from when the symptoms starts is when they die? Like,
I don't know. I don't know how long it lasts. Maybe, but I'm not sure.
yeah, like I read an article of like rabies being like the most deadliest,
Oh, yeah.
dangerous, like
Oh, yeah.
bacteria that, like,
It's not bacteria.
I know. Bacteria.
The virus, yeah. If you start showing symptoms, write that will, because you're not going to make it. 10 people in forever, your chances aren't so good. So I think there's been a couple of advances lately. The last couple cases, I think what they've done is they've put them in an induced coma, and they've been able to do something, I don't know exactly what, but I think they are making some advances. And so the number might now be a little higher than 10, but it's still not, the odds are not in your favor. So, and back during, you guys with me? Back during Pasteur's time, the only thing they could do for you, because they didn't have that preventative vaccine that we have that will prevent it from happening, the only thing they could do to potentially save an individual's life was cauterize the wound with a hot poker or something, in hopes that they got it soon enough that by cauterizing the wound, that heat just kills off that virus before it starts to spread up the nerves, right? That can't be pleasant, right? That can't be fun. So he started working on a vaccine against rabies. And he used the dried spinal cord from rabbits. And I like to say he used it from rabid rabbits, right? So rabbits that had died of rabies, he dried their spinal cords and ground it into a powder and used that for the vaccine, right? So he was working on it, he was testing animals, and it was working really well. But he hadn't tested it on humans. And a woman came to him with her 9-year-old son. His name was Joseph Meister, you don't need to know his name. And she said, please, 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 I know you're working on this vaccine. Can you please use it on my son? He was attacked by a rabid dog. And the little boy had bites all over. You couldn't cauterize all those wounds. It was just too much. And so he was skeptical. He didn't know if he should do it or not because he hadn't tested on humans. But this boy was going to die. no matter what. If he didn't try it, the boy was going to die. If he tried it and it didn't work, the boy was going to die. So what's the harm in trying it? So he did try it on the little boy, and it worked, and it saved the little boy's life. And it became the first rabies vaccine. Now, the boy didn't have clinical symptoms. He just had the attack, okay? Interesting story, the little boy grew up and became the security guard at the Pasteur Institute to honor the man that saved his life. And then the story gets kind of sad after that, because he ended up committing suicide, and there's a couple different variations on the story. It's kind of like urban myths and urban legends. There's a lot of them in science too. So some people say, well, you know, when Germany marched into Paris, he didn't want the Germans to desecrate Pasteur's tomb, and so he killed himself. Other stories say it had nothing to do with Pasteur. He was having personal issues at home, and either way, it didn't end well. But he was the first to receive the vaccine, and that was a good thing. So those were Louis Pasteur's successes. So we'll move on and get started talking about Robert Koch. He was a German, and he was a contemporary of Louis Pasteur, meaning he was from roughly the same time period. And they did a lot of similar things. They worked with a lot of similar organisms and so forth. So some of the things that he did, he identified the cause of anthrax as Bacillus anthracis. We know that Louis Pasteur worked with Bacillus anthracis, so there were some things they had in common. He also recognized the difference between active or vegetative cells versus bacterial endospores. So most bacteria are what we call vegetative. In other words, the cells grow and divide, grow and divide, grow and divide. That's pretty much their life. It's kind of boring, but that's what they do. We call them vegetative cells. Okay? However, there's some bacteria that have this ability to form what are called endospores. And they do this when the environmental conditions are not conducive to survival. So with a normal vegetative cell, if maybe there's no moisture or no nutrients, or maybe there's radiation or some sort of adverse environmental condition, the vegetative cells die. Okay? They die. But if they can produce endospores, and we'll talk more about these later this semester, what happens is those vegetative cells of certain species will take their genetic material and wrap all these protective layers around it, and then the cell itself disintegrates, and all you're left with is this spore, and it's the genetic material coated in a lot of these different strong layers that protect it. Protects it from heat, lack of moisture, radiation, harsh chemicals, heat, cold, you name it, it protects it. It's almost like a state of dormancy. These endospores can survive millions of years, and then when the conditions become appropriate in the environment again, they undergo what's called germination, and then they revert back into vegetative cells. Okay? Good example of this was back in 2001 when the Bacillus anthracis spores were being sent through the mail. Anyone alive then? You guys are young. You guys are young. So what was happening was they had these endospores in the mail. People would open their mail, and what would happen? It was kind of like a powder. They'd breathe it in, right? Endospores themselves can do no harm, but they breathe them in, and they go into the lungs, right? It's warm in your lungs, it's moist in your lungs, there's nutrients in there for the organism. So guess what? Those endospores underwent germination, became vegetative cells, actively dividing cells. Those are the ones that can cause disease. And so that's what was happening with the anthrax problem of 2001. 2001. Those endospores themselves can't do anything, but when they make their way into that nice, warm, moist environment, now they can germinate and become vegetative cells. So he was the first to identify that things can do this. Now, two different genus of organisms that can produce endospores, and you will need to know these: Bacillus, so things like Bacillus anthracis, right? And I just put an SP next to it. That just means all species of Bacillus. So there's lots of species: Bacillus anthracis, Bacillus cereus, Bacillus subtilis. There's a whole slew of different Bacillus species. So if I put SP, it just means all of that genus, all of the species within that group. Okay? Another one would be Clostridium. And again, I'm going to put that SP next to it. So all of the organisms within that genus can produce endospores. You may have heard of some of them. So things like Clostridium tetani, which causes tetanus. Clostridium botulinum, which causes botulism. Clostridium difficile. Anyone ever hear of that one? If you work in a hospital, you have. That's good old C. diff, right? It's lower than that. It's lower than that. It's
Doesn't hand sanitizer not touch that?
probably not because of those endospores. Yeah, it's a tough one to get rid of. I mean, what do they have to do to a room after someone's had it? A lot of work, because those endospores are resistant to a lot of stuff, and they're little. They're even smaller than the cells would be, so they're really hard to get rid of, and it's a nightmare, I'm sure. Plus, from what I understand, it's not a fun thing to have. I've known people who've had it, and they've said they wouldn't wish it on their worst enemy. So those are some examples. Other things that he did, he identified the cause of tuberculosis as a bacterium called Mycobacterium tuberculosis. He did a couple things with this organism. This organism has, most bacteria have a cell wall. This one does too, but it has a lot of wax in the cell wall. It has a special layer in the cell wall that's very waxy. It protects the organism. Think about what chapstick does to your lips. It protects it. So does the wax in the cell wall of Mycobacterium tuberculosis. With that being said, it makes it very difficult to stain it, to view under the microscope, because the stain can't get past that waxy layer. So he actually developed a staining technique that would help you see that organism. So that was really important. Also, he tried to make a vaccine for this organism using a protein derived from the organism called tuberculin. It didn't make a good vaccine. Some substances elicit a strong immune response from an individual. Some substances don't. Just because it's from the microbe doesn't mean it's going to be a good substance for making a vaccine. That's why there's a lot of research when designing a vaccine, because not everything is going to make your immune system mount a strong response. This did not. So it did not make for a vaccine. However, it's still used today when doing like a PPD test. It's the protein that's injected under your skin in that test. And so it's used to detect the presence of Mycobacterium tuberculosis, but it's not able to be used as a vaccine. There is a vaccine, but it's not this particular protein. And then the last thing I wanted to mention today is that he also was able to grow bacteria on plates, just kind of like you did, right? He'd grow bacteria on plates, he'd spread the organism thin, and it would grow into these nice colonies. But he was having trouble because he was using gelatin, okay? Now, we like to incubate our bacteria between 35 and 37 Celsius. That's human body temp, okay? If you were to take gelatin, Jell-O, and hold it tight in your hand, what would happen? It would melt. So if you were to put plates made of gelatin, gelatin was in the plastic plate. Well, it wasn't a plastic plate back then, it was glass plate. But if you were to put gelatin in there and then put it in the incubator, you'd have a mess. So it was the wife of one of his coworkers, and I finally got her name down. I've always just called her the wife of his coworker. Her name was Angelina Heiss, Heiss, who came to him and said, you know, instead of using gelatin, you could use this thing called agar. Agar is a protein that's very similar to gelatin. Gelatin is animal-based, an animal-based protein. Agar is an algae-based protein. But they have similar properties in that they get that gel-like consistency. The only thing is agar melts at a higher temperature. So you can put it in an incubator at 35 to 37, and it's not going to turn into a gooey, melted mess. So she gets credit for that. So that's where we're going to finish today, and we'll continue with Robert Koch tomorrow and or next class, and talk about his postulates. Okay? Just keep in mind, we'll finish all that up on Thursday.